And exams are up here. What's your exam? Yeah, you could put it alphabetical, or you could just like flip it over and throw it in a separate pile. Okay. You know, then I know people have looked at it. Like, these should be sort of alphabetical. They were alphabetical. At some point, they were. Good. Okay. Did it start as it is? Oh, it did. Okay. And exams are over here if you want to see your exam. I feel like, oh, I am missing something. I knew I was missing something. Happy Friday, y'all. That's so quiet. Yeah, me too. But I'm so excited because tomorrow Charles is, well, we are going to pick fish. We actually are going to pick fish, and he's actually starting to get excited about it. But I do not get to sleep in tomorrow morning because he's taking his rally test for piano at 8:15 in the morning on campus. Here? Yes. So.
What is that? Oh, like literary rally?
No, it's like a test for music. Like, so he goes to piano, like he goes to piano class, you know, like, and knows more about piano than I do. A lot more. And so they have festival where they have to play. They have rally where they have to take a test. Then they have a second rally where they have to play and sight read. Like they play something they already know and then they sight read. And then there's
how old is
recital. 9.
That's a lot of work for a 9-year-old.
He's really good at it though. He's really good at it. It is a lot of work though. It's okay.
The ones that I missed, like I thought I was.
Exams are over here if you want your exam. Go see your exam.
Not all of them, but the ones that you missed. Yeah.
I'll do the same thing. I feel like I don't.
It happens. So I do not get to sleep in tomorrow. Sunday, I am sleeping in. That's fun. Oh.
So anyway, that's 2 hours away.
That's 2 hours. Yeah, you're not sleeping in. No. Do your attendance.
Guys, and I don't even have a kid. Just life.
It's just life. But like one day, I just want one day a week where I can sleep past like 7 a.m.
But doesn't your body like do that thing where you just like wake up?
I wake up and go to the bathroom and I go back to bed.
See, once I'm up.
Oh no, I can go back to bed. Absolutely, I can go back to bed. All right, y'all ready? So I have a couple of jokes for y'all today. Okay, this is the science nerd joke. What did the limestone say to the geologist? Make sure I get this right. Wait, now I can't remember the punchline. Oh, this is terrible. Oh, don't take me for granted. Don't take me for granted. It's terrible when you can't even remember the punchline. There's so many jokes. Okay, this is an easier one to remember. What do you call a cow with no legs? Ground beef. Ground beef. Charles really likes that one. All right, so make sure you do your attendance. Please, please do your attendance. All right. And we were talking about different catalytic mechanisms, right? And we said one of them was acid-base catalysis, and there were two different ways that it could be acid-base catalysis. It could be general and it could be specific. If it is specific, what molecule are we talking about? If it is general, what are we talking about? Amino acid side chains, right? Amino acid side chains. Okay. So we talked about that one. Covalent catalysis. What do you think we're going to do in covalent catalysis? We're going to make some bonds, right? We're going to make some bonds. Okay, so in this example, we have a nucleophile that is actually going to work on our substrate, right? So here's our enzyme, right? Look what we have over here. What is this? Cysteine, right? Okay. And then what is this? What is NAD plus? Not like what does it stand for, but what general classification of molecules? A cofactor. A cofactor. How do you know it's a cofactor and not something else? It's organic. If it was inorganic, it would be no. Yeah. Okay. So watch. Go back real quick. Where was that? How far back was that? Here, right? Cofactor is going to be inorganic. Coenzyme is going to be organic. So what is it? It's a coenzyme. It's a coenzyme. Okay. It's not a metal. Where were we? Okay, here we go. All right. So we have our NAD+. That's our coenzyme. Our coenzyme is going to participate in this reaction. So the first thing that happens is your substrate comes into this pocket, right? It's always going to be a particular place. And so you're going to get formation of an intermediate. So before the NAD+ participates in the reaction, we're going to have a reaction with a side chain in the enzyme itself. And we're going to make an intermediate, a thiohemiacetal intermediate. So let's just say I wanted to design a analog that would be an inhibitor of this particular enzyme. What am I worried about? Am I worried about this R group? No. What do I want to make sure I have? A carbon that is in what shape? A tetrahedral shape, right? So this is an intermediate that you could mimic when you're making your analog and making your inhibitors. Okay, so your substrate binds, right? What happens when your substrate binds? You go from being, what is that? Trigonal planar. Now we go into tetrahedral, right? So what happens here is that now our NAD+ can function to accept this hydrogen. When it accepts the hydrogen, is the carbon happy? No, what's going to happen? Lone pair on the oxygen goes and makes a double bond. So now we have a carbonyl carbon, right? So our coenzyme NAD+ functions as an oxidizer, right? So now we have NADH. Can't function that way in the enzyme anymore. So the enzyme basically kicks it out and then picks up a new NAD+. Okay? So now what happens is the whole point of this is to go through and actually phosphorylate a substrate, right? So we want to attach a phosphoryl group to a carbon. Well, all we did was prepare our substrate to do that. So we needed to change the molecule so that it would be easier to accept a phosphate group, right? Just attaching a phosphate group to our substrate is highly energetically unfavorable, right? But if we can change it and turn it into an intermediate where that reaction is more favorable, that's the whole point of an enzyme. Now we have a substrate that's easier to phosphorylate. And then once that phosphorylation event occurs, right, we have nucleophilic attack of this oxygen on the phosphate group for that carbonyl carbon, and we can form our phosphorylated product, right? So that's the whole point of an enzyme, is that sometimes it's not going to take exactly the same path as the uncatalyzed reaction. Sometimes we're going to make new substrates in order to facilitate an easier 2 steps instead of a difficult one step, right? Okay. So you can also have metal ion catalysis, right? So in metal ion catalysis, you can have, remember our whole list of different metal ions that are possible? This is just one of them. This is zinc. So a lot of times there are multiple functions of your metal ions inside of an enzyme. One of them is that it will properly orient your substrate molecule inside the pocket, right? Because if you're positively charged, what can you then attract? Something that's negatively charged. And so you can make sure that a certain part of the substrate molecule fits inside of the pocket at a particular place. But it can also aid in an actual redox reaction. So it's really multifold, the function of an ion inside of a enzyme. So in this case, right, we have Zn2+. So what's going to happen to this one, right, is we have this hydroxyl group here attached to our zinc. So this hydroxyl group, the oxygen is going to attack our carbon, right? We're going to form a new bond between the oxygen and the carbon. When we do that, what happens to the one of the double bonds? It really doesn't matter which one. What's going to happen? We're going to lose the double bond and turn it into a lone pair. So now this oxygen is carrying the negative charge, right? Now, what happens then in the next step requires the presence of water. This is not general catalysis. This is not, I'm sorry, this is not specific catalysis because the water is not doing the catalytic activity. It's required for the function, but it is not doing the catalytic activity. So you don't call it that. But in the presence of water, water gets cleaved, and what do you end up with? Your hydroxyl group, right, is going to go on the substrate and is going to get kicked out. You have one hydroxyl group from the water that's now going to be attached to the zinc, and then you'll have a free hydrogen ion. So what did we start out with? What substrate did we start out with here? Carbon dioxide. What did we make? We made bicarbonate and a proton. We made an acidic environment. Where is this important? Where do we make carbon dioxide? In all of our cells, right? Where does the carbon dioxide go? Into the bloodstream. What happens to carbon dioxide when it's in the bloodstream? Gets converted to bicarbonate and a hydrogen ion, right? So this is a way that your body can actually regulate pH inside of your bloodstream, right? All right, so when we talk about our reactions, we talked about mechanisms, right? We talked about the different kinds of mechanisms. And now we're going to kind of talk about the different types of reactions. So we have a lot of reactions inside of our body, a lot that are redox reactions. So if you think about the different kinds of reactions that are going to use redox, so this would be glycolysis. What else might use redox reactions? Once you go through glycolysis, what's your next step? Where are you going next generally? Citrate. Citrate uses lots of redox reactions. Where are you going next? Electron transport chain. Now, if you're a plant and you can go backwards, right? What kind of a reaction are you doing? What general, like, you know, we've listed all these different things, but what do we call it in plants if you're going backwards? Photosynthesis. So lots of different reactions inside of our bodies are redox reactions. So if you didn't have redox reactions, none of that stuff would go, and it would be very, very, very bad. So you have, all right. So you have different molecules that are electron carrying molecules, and they come in pairs, right? So what's the difference between NAD plus and NADH? Hydrogen. So yes, this portion of the molecule is important, but a lot of times we don't draw the structure of NAD and NAD NAD plus and NADH, because all we really care about is that one hydrogen. So you'll see that a lot in biochemistry. Like NAD is kind of a big molecule, relatively. But we really only care about the hydrogen. So we don't really worry about drawing anything else. We draw things like R groups. Oh, there's a big honking R group on there. But what do we really care about is the hydrogen. So same thing with NADP plus and NADPH, which you'll find out where we use these different reactions, these different coenzymes. FAD plus, FADH2, right? What do you notice? What's different between all of them? Hydrogens, right? So what kind of a reaction are we doing? Redox, redox. One example, did we talk about this in this class? Lactate dehydrogenase, all runs together for me. Yes, all right, and we're actually gonna do a protein purification next lab. It's protein purification, love my protein purification of lactate dehydrogenase from hopefully beef heart. Hoping for beef heart, we will see, where we convert pyruvate into lactate. So what happens to pyruvate when we convert to lactate? Look at the structure, what did you gain? You gained 2 hydrogens. What happened to NADH? You lost. So that's an oxidation reduction reaction. There are lots of reactions like this inside of the cell. Here we're going from succinate to fumarate, right? What do we a lot of times call these enzymes that perform these redox reactions? Dehydrogenases, right? Because you're removing hydrogens. So just kind of get familiar with what we call the enzymes that do particular reactions. And there was a table from last time, I do believe, pretty sure. Okay. So you can also have metabolic transformation reactions. So in transformation reactions, you have a lot of isomerizations where you are just changing how things are arranged. Like here, this is an isomerization. Because are you changing your number of atoms? No. All we're doing is moving those hydrogens around and then adjusting the bonds, right? You can also have condensation reactions. Condensation reactions, what happens in a general condensation reaction? You're going to make water. And what happens when you make that water? Like you get a hydrogen from one group, you get a hydroxyl from another group, right? And it's attached to R, say R1, R2, right? And so what happens? You put R1 and R2 together. So whenever you're combining 2 things, then you make water as a byproduct. All right. You can also have all kinds of different reversible covalent modifications. What is the #1 reversible the most common covalent post-translational modification we've talked about so far? Phosphorylation. Absolutely. Phosphorylation occurs on proteins all the time. And that's our molecular on-off switch. Phosphorylate it, it's active. Dephosphorylate it, it's inactive. And vice versa, right? And so that's good for protein activity. But sometimes I don't even want to make the protein. How do I not make the protein? What am I going to have to change? Now you could change the transcription factor. And sometimes that's how it works, right? Your transcription factor is inactive until a hormone comes in and binds to it, and then it transcribes the gene. That's one way that it could happen. But there's another way that it could happen where we don't actually modify the transcription factor. What else could we modify? DNA. We can modify DNA. Have you heard about methylation of DNA? What happens when we methylate DNA? So phosphorylation, methylation. Methylation is usually DNA. This is phosphorylation is usually protein. What happens when we methylate DNA? We silence it. It's a silencing activity. So when you methylate, basically those methyl groups go in the major groove of the DNA. And what normally fits in the major groove of DNA? all of the different enzymes that are required to make mRNA. That's where they fit. So you're basically excluding them and making sure that they can't work. So a lot of times in this class, we'll talk about the protein, but I kind of do want you to know that there are ways that we can modify the DNA and have an effect on cellular responses and things like that. So for our phosphorylation events, right? If you're going to add the phosphate, that's a kinase. If you remove the phosphate, that's a phosphatase. So where do you get the phosphoryl group from this? Typically, you get it from ATP, typically, right? So if you phosphorylate any given enzyme, you can make it active or you can make it deactive, right? So here's a kinase that is dependent upon ATP. And so if you have ATP present, this enzyme is able to modify its substrate, put a phosphoryl group on it, and now we can turn on insulin signaling, right? So if you have the phosphoryl group in this case, now it's on. Could be either way. Doesn't really matter. All right. So we talked about mechanisms. We talked about different classes of reactions. Now we really have to talk about the kinetics of reactions. This is a really, really, really cool section. And it's really important to understand because in order to understand how an enzyme works in the body and how you're going to regulate activity of all kinds of different processes that occur in the cell, you have to understand kinetics. You have to understand how efficient something is, how substrate concentration will affect it, how temperature will affect it, how all kinds of things are going to affect it. So we'll talk about some basics and then we'll kind of put it all together. So we already know that our substrates are going to bind to our enzymes at the active site, right? And that initial interaction is pretty weak, right? Then they're going to have stronger interactions with the intermediate. That intermediate is going to go to a product, and then the product's going to get kicked out, right? So all of the catalytic reaction mechanisms that we're going to describe are all organic chemistry. So if you did well in organic chemistry, this stuff is all going to make sense to you. So let's take an example one at a time. If organic chemistry didn't make sense to you, I'm sorry. So chemotrypsin is a serine protease. So what do you think is going to be the catalytic side chain that is going to facilitate our change? Serine. It's going to be serine, right? So we have both a covalent and an acid-base catalysis that are going to happen. Now, a lot of proteases have a similar mechanism, and that mechanism is called a catalytic triad. So we have, what's this one? Histidine, asparagine, serine. So, and these different residues form hydrogen bonds. And it's the changing of hydrogen bonds with the amino acid side chains and the substrate that allow for the catalysis to happen. Okay? Yeah. Oh, you're right. Did I say, what did I say? Oh, aspartate. Thank you, aspartate. Sorry. All right. So when we look at these three, we're looking at three different residues. So if the enzyme is empty, if there is no substrate inside of that enzyme, these residues will interact with one another through hydrogen bonding. So what we can see here is that we have an initial hydrogen bond between oxygen and hydrogen coming off of our nitrogen right here, right? Okay, so what's going to happen is in this histidine, we have two nitrogens. One of the nitrogens has a lone pair on it. That lone pair, if it's bonded to, if you, instead of have a lone pair, if you bond it to a hydrogen, what happens to the charge on the nitrogen? It becomes positive. So when we look at this, right, if this nitrogen becomes positive, what happens to our serine? What happens to the oxygen on the serine? it becomes negative. So we stole the hydrogen, right, from serine and gave it to histidine. So when you look at our catalytic mechanism, and I really want to be able to zoom in, and I forgot how to do that. I think I can zoom in this way. Zoom. Ooh, not enough. Okay. Very nice, much better. Okay, so inside of our enzyme, right? This is chemotrypsin. So chemotrypsin cleaves very, very specific residues in a peptide chain, right? If you eat protein, chemotrypsin is one of those enzymes that's going to digest that dietary protein. So each enzyme, there are many, many enzymes that will cleave proteins that are proteases, but they have very specific pockets within the active site. In this case, what kind of a side chain are we binding? An aromatic, right? An aromatic side chain. Okay, so here's our peptide substrate, and this peptide substrate has an aromatic residue on it. When this peptide fits into the pocket, the aromatic residue will fit into a specificity pocket, which will then orient our carbonyl, the oxygen on the carbonyl carbon, in close association with serine 195 and glycine 193. What do they both have in common? A nitrogen with a hydrogen attached to it, right? Okay, so this is what's called the oxyanion hole. And so this one provides stability to this, what's going to be a negatively charged oxygen in just a second, right? Okay, so when the peptide bonds, right, the next step here is, and I hate that they took away, oh my gosh, that's so large, right? But here's our His residue. So the His residue right here is going to steal away that hydrogen. When that hydrogen is stolen, what happens here? Now we have an oxygen that is going to attack our carbon in the carbonyl. What happens when that occurs? We're going to form this really, really weird tetrahedral intermediate. This tetrahedral intermediate, right, has lost the double bond between the carbon and the oxygen. Can y'all see that? Now, what is that? What's the charge on the oxygen? Negative. So now we have a stabilization of that negatively charged oxygen. This is the whole point of an enzyme. Is that a stable intermediate? Is that something that is going to form outside of an enzyme? No, but we have this really cool local environment that is going to provide stability to this really weird intermediate. So it'll be really, really easy for this intermediate to form, right? Okay, so that's our first intermediate, right? So once that intermediate forms, then, right, we're going to have a few other things happen. So once our anion is stabilized, what's going to happen is that we'll have peptide bond cleavage. So right here, this is our peptide bond right there. That's the part that we want to cleave. We want to be able to remove one residue, right? Cleave between the two residues. So it's between the carbon and the nitrogen where we're going to make that bond. So His is going to steal back its hydrogen. When histidine steals back its hydrogen, this bond between the carbon and the nitrogen is going to become broken. So when we do that, now we can release one peptide fragment, but we have another peptide fragment, right? So now what's going to happen? Our His is going to donate our proton to serine 195. So once we donate that proton to serine 195, right, we're going to make a second tetrahedral intermediate. What's that?
Where does that water
Which on the left side here?
Yes.
So water, it's just water.
So it's just
You're in an aqueous environment unless your pocket is specifically excluding water. Some do. Some enzymatic pockets exclude water and some do not. Some let water in, but water's everywhere. So unless you specifically exclude it, it's not going anywhere.
I know it came from the first product leaving
Well, you have room now. You have room for the water. It's a very small area. So you can't have everything in there at once. So once your one fragment comes off, then you kind of have room for water, but it's not specifically recruited in that way. It's just always kind of sitting there and waiting. Okay. All right. Okay. So now your histidine is going to steal another hydrogen back. It steals the hydrogen from water. What are you left with? Hydroxide, right? That hydroxide, the oxygen is going to attack the carbon. It's going to make a new tetrahedral intermediate. So both intermediates are tetrahedral. So you could make an analog that's tetrahedral, right? So again, at the same time, your oxyanion of that intermediate is stabilized inside of the pocket. And then the last step, right, your His 57 is going to donate that proton to serine 195. And then we're going to release our second peptide fragment. And now, what do you notice about this arrangement compared to this arrangement? I know it's hard to see because I have to scroll, but it's exactly the same. This is really, really important. Why is this really important? We can go again. We can go all over again, right? All right. Do you have to memorize that whole thing though? You don't have to memorize the whole mechanism. You have to be able to explain it if I show you a picture, but you don't have to memorize the whole mechanism, right? We're going to go through quite a few different ones, so you don't get lost in the weeds, is what I'm saying. That makes sense. All right, so here's chemotrypsin, right? This is a very, very simplified version of chemotrypsin. So you're making sure that this pocket can accommodate a huge ring structure, right? That's a really big ring structure. If you have a second enzyme that cleaves proteins, right? But let's just say I want it to cleave at positively charged residues. What's going to be in my pocket? Negatively charged residues, right? So if you can actually look at the structure and the arrangement of side chains within the enzymatic pockets, you can figure out what the substrates are. Elastase is an enzyme that cleaves elastin, which is a protein, right? And elastin has tons and tons of glycines and alanines, right? Really, really small residues. So look at the size of the pocket comparatively. It's really small. We have threonine and valine that are sucking up all the volume inside of the pocket. So it's still going to cleave a protein, but it's going to cleave a protein that has much, much smaller side chain residues, right? So you can look at a pocket and you can kind of make predictions on what it's going to be able to cut. All right. So this is another class of enzymes, a metalloenzyme. So what's a metalloenzyme? It has metal ions inside of it that never really leave. A metalloenzyme, those metal ions are basically permanently attached to the enzyme. So the one we're going to talk about is Inolase, and this is a glycolytic enzyme. And it's actually used in gluconeogenesis as well, so it can catalyze both the forward and reverse. So inside of, it's a dimer. Thank you, a dimer, where inside of your active site, you have a magnesium 2+ ion. So you have two active sites because it's a dimer. And then this little peptide in here, this is like a possible product, like an analog. Okay, so if we zoom in to Inolase, what do you notice that's surrounding Mg2+? What do we have here? Negative charges. We have negative charges, because we want to keep this magnesium stable, because it's going to be really, really, really important in our catalytic mechanism, right? So if we look at our catalytic mechanism, right, in this, we're going to have some residues that are going to act as a general base. So if it's a base, what is it going to do? It's going to take protons. So we're going to take this proton right here that's on our substrate, and we're going to pull it onto the nitrogen. So what happens to that carbon right there? We're going to end up with a double bond. But what's attached to the first carbon? This is carbon 2, this is carbon 1. I have 2 negatively charged oxygens. Is that a very stable configuration? Absolutely not. But why is it possible to have this really, really, really weird intermediate? Because we're stabilized by the positive charges on the magnesium. So all of the enzymes that we talk about have structures that facilitate the ability of those high energy intermediates to form, right? Okay, now what do we have? We have a general acid going on because we're going to steal away what? A hydrogen here. We're going to steal this hydrogen right here. And it's going to go and attach to our substrate, right? Now in the presence, what are we going to produce? We're going to produce water. We're going to produce water. And then we're going to make our phosphoenol pyruvate product. Now this does not show the regeneration. This is just the enzymatic activity, which is okay. All right. The next class are kind of, these are ones that contain those oxidation reduction coenzymes that are really important. So one of the ones we're going to use as an example is HMG-CoA reductase. So you know, if people have high cholesterol, they prescribe you a drug. We talked a little bit about this, right? A statin. Did we say this? So what does a statin do? A statin actually binds to HMG-CoA reductase and inhibits the ability of HMG-CoA reductase to work. So if this enzyme is involved in cholesterol biosynthesis, how is that going to reduce your cholesterol levels? You're not synthesizing it anymore. There's 2 ways to get cholesterol. You eat it or you make it. It's not an essential lipid. It's not something we can't make. We can actually make it. We actually make more than our bodies can absorb in a day. We make more. So if you're going to inhibit a process, you want to inhibit synthesis so that you can reduce somebody's cholesterol levels. And so what kind of a target, how are you going to target this enzyme? Find its binding site. What does its intermediate look like, right? All that kind of stuff. So in order to understand that, in order to design drugs that are effective against whatever you're trying to do, you have to understand how it all fits together, right? So this one has 4 binding sites because you have 4 subunits, right? And each one has a cofactor, NADPH. So in this one, what we want to do is we have this HMG-CoA, which has coenzyme A attached to it. And what we really want to do is to remove the coenzyme A. That's our whole goal of this reaction. So kind of keep that in mind when you're looking at all the different things that happen. There's got to be an easier way to do this, but that's okay. All right, so here we've got HMG-CoA. This is our substrate.
So is our test going to be on all mechanisms?
Some? It's going to be on some mechanisms. Yes. What?
And painful.
It's not on all mechanisms, but it's on some mechanisms. Yes.
So we basically have.
So don't memorize them.
Right,
We're
but
gonna
we need to know how to explain every single one of these just in case.
You do have to be able to explain them.
memorize them, like you said.
Don't memorize them. You don't have to memorize them.
You have to know them well
But you
enough
have to
know your
understand
opinion.
them enough.
Yes, you have to understand them enough. Yes.
Okay,
Yes.
all of them. Right?
Should have said memorize when we do.
Ah, all right, so you got HMG-CoA, right? We want to get rid of this coenzyme A. So what's going to happen is we're going to have one residue that's going to act as a general acid. What's a general acid going to do? Deprotonate, right? Okay, so if we come over here, what's happened? What's changed? We've pulled a hydrogen off of NADPH and attached it to our intermediate, right? When we do that, what did we lose? the double bond, and we gained our hydrogen right here, right? So now we have a positively charged residue here that is going to stabilize these two oxygens here and this oxygen here, right? So we're going to stabilize all those negative charges. So now we're going to exchange NADPH for NADP plus, right? Because now we've used that NADPH and now we've made NADP plus. So we're going to get rid of that and we're going to get a new NADPH. All right. So what happens next is that our His right here is going to donate this proton right here. So if we come down, it's so hard to show the whole thing. We also have, if we're going to pick up this hydrogen right here, what else is going to happen? Look over here. We're going to pull this hydrogen off because we're going to act as a general base, and you're going to end up with a double bond oxygen here. So remember, the whole point is to get rid of our CoA. So when that happens, right, we're going to have a hydride transfer from NADPH. So now we have that double bond here. What bond did we break? Carbon sulfur bond right there. That's what we broke. And that's the whole point, right? We're trying to get this CoA out. So now we're going to have another general acid event, and we're going to pull that hydrogen off, put it back onto our original substrate, and we have another hydrogen from NADPH. So now we've made basically our two substrates, CoA that can go and be released, and then melavonate is our other substrate. But the whole goal was to get the coenzyme A off, right? Okay. That's it for mechanisms, if that makes you feel any better. No, not really. It doesn't. Okay, let's see. No, I want to do full screen. Okay. All right. So now that you kind of understand what has to happen inside of an enzyme pocket, now we can talk about kinetics. Now we can talk about how fast something happens, right? So remember, your delta G for any given reaction is not going to change, right? But if we add an enzyme to it, what do we do to the rate of the reaction? We increase the rate of the reaction. So a lot of times we reference rates of reactions as velocity. So you'll see it as V. So we're going to use that today. You also need to be able to identify substrate concentration. So we put it in brackets and we put an S, and that's generic for substrate. Little k, right? I try to write the little k as a little looped one. You know, it's not big K. You know, there's a difference between big K and little k. Yes, okay. So the velocity of a reaction is dependent upon the rate constant of your reaction times the substrate concentration, if this is what kind of a reaction? A first order reaction. So we've said that this is what an enzyme will do. You'll have an enzyme in the presence of a substrate that will make an enzyme substrate complex. That substrate will turn into a product, and then the product will get kicked out. So when you look at this reaction, honestly, this is actually a double-headed arrow. It can go backwards once you get a product, but is it very likely? No, it's very, 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 very unlikely. So we call this K-2, and we say K-2 is negligible. NEG, something like that, negligible. So when we talk about the kinetics of a reaction, we really kind of don't worry about K-2. We say that not enough of the product will actually revert back to substrate, because it doesn't, okay? So we're going to look at three different things. K1 is formation of ES, right? K-1 is when our enzyme substrate breaks down. K2 is when our enzyme substrate converts to enzyme plus product. So whether you're talking about K2 or K to the negative 1, K negative 1, both of these are talking about what general function happening. Enzyme substrate what? ES breaking up, right? Right? What is K1? Enzyme substrate formation. That's going to be really important. Okay. All right. So, how do we measure velocity of a reaction? I'm gonna draw something else first. If I have a reaction and I add just a little bit of substrate, if this is my reaction progression and this is concentration of substrate, what's going to happen to the rate of my reaction? If I have just a little bit of substrate, it's going to go pretty slow, right? What happens if I increase the amount of substrate? It's going to go faster. What if I increase again? Faster. What if I increase again? Faster. What if I increase again? A little bit faster. What if I increase again? I'm going to get to a point where just increasing is not going to change the rate of the reaction, right? So that's what we've graphed down here on the second chart down there, right? Initial substrate concentration versus velocity. So if I look at how fast the reaction is going, there's going to reach a limit of substrate concentration, right? When I get above right about here, I can increase my substrate concentration hugely. And am I going to have an effect on the rate of the reaction? No, absolutely not. But here where I have a relatively low concentration of substrate, I can have generally a big effect on the reaction, right? If I double the amount of my substrate concentration in this area, what's going to happen to my rate? It's going to double, right? That's a first order rate reaction. If I double my substrate concentration, I double my reaction speed, right? Okay, so the velocity of our reaction can vary with our substrate concentration. We agree on that? Right. So when the substrate concentration is sufficiently high, then the reaction is said to have reached the maximum velocity. So right here, this is our max velocity, or V max. That's saying I can't add any more substrate. That's as fast as my reaction can go. Yeah? Okay. Vmax is right here. Like, let me erase some of this stuff. It kind of got crazy, didn't it? Okay. So this right here, if you were to draw where it becomes linear, this is Vmax. Yeah? Okay. All right. So now that you kind of understand that, I want to show you an animation of kinetics, and then we're going to talk about it in a little bit more detail. Here we go. Let's see if this works. Come on.
Of an enzyme is how scientists characterize and compare enzyme catalyzed reactions. The overall enzyme reaction can be represented by the following formula, with the enzyme E and substrate S reversibly associating, reacting, and rapidly dissociating into the enzyme and product P. If we graph the concentration of enzyme substrate complex, and product over time, we would see an initial burst of product formation that corresponds directly to the first step of the reaction, enzyme substrate complex formation. This is called the pre-steady state period. After the pre-steady state period, The reaction reaches steady state, where all enzyme reaction steps are occurring at a steady rate. This is the steady state period, and during this period, the substrate concentration declines steadily as product is formed. By plotting the change in product concentration during the steady state period and finding the slope of the line, we can calculate the initial velocity of the reaction. Initial velocities are important for finding Michaelis-Menten parameters. When we determine the initial reaction velocities for different starting substrate concentrations, we see two trends. In these experiments, the enzyme concentration and the volume of the reaction are both held constant, whereas the substrate concentration is varied. First, the higher the initial substrate concentration is, the higher the initial velocity. Second, although initial velocities increase, the increase is not as great at higher substrate concentrations than at lower ones, because at high concentration, we are beginning to saturate the binding sites of the enzymes in solution.
So
For
that's
this reason, when
that's really, really important. When you're up here at really high substrate concentrations, that means that every single enzyme is bound and is doing its job. Down here, do you have every single enzyme bound? No, you have lots of free enzyme. It's really important.
we plot initial velocity versus initial substrate concentration, we get a hyperbolic curve that is characteristic of first-order Michaelis-Menten kinetics. Using this plot, we can now determine other important Michaelis-Menten kinetic parameters. The velocity that the curve approaches asymptotically is the maximum possible velocity of the reaction, or Vmax. If we divide the Vmax by 2 to find half the maximum velocity, the corresponding initial substrate concentration is known as the Michaelis constant, or Km. Increasing or decreasing the enzyme concentration will change the maximum possible velocity, but will not change the Michaelis constant. It's a constant property of the enzyme reaction. Another way to look at the same data is to plot the reciprocal of the initial velocities and substrate concentration. This converts the data into straight lines and is known as the Lineweaver-Burke plot. Lineweaver-Burke plots make it easier to obtain the Michaelis-Menten parameters. The y-intercept corresponds to the reciprocal of Vmax, the x-intercept to the negative reciprocal of Km, and the slope is the ratio of Km to Vmax. Another way to compare enzymes is to determine the maximum catalytic activity under saturating levels of substrate. This value, called the turnover number, is defined as kcat. The kcat for a reaction describes the catalytic rate, but it does not reveal the catalytic efficiency. In the case of the enzyme fumarase, the Km for fumarate is 5 times lower than it is for malate, yet the kcat values for these two substrates are nearly the same. This results in a specificity constant for fumarate that is about 4 times higher for the enzyme than when malate is the substrate. This higher efficiency means that fumarate is the preferred substrate of the enzyme.
Okay. So we're going to talk about each of those individual properties one at a time, but how much time do you have? 5 minutes. Okay. So it's important to understand that we can't monitor reactions outside of steady state. It's really, really difficult to monitor those conditions. So basically everything that we're going to talk about is steady state. And so we're going to convert these values, and we're going to talk about two equations. Now, these equations you have to memorize. They're on the formula sheet. They're on the formula sheet. So don't get too crazy about it. So remember our equation. Remember what happened? We had enzyme plus substrate goes to what? ES. And then we had a single arrow, and that single arrow goes to enzyme plus product. Okay, so what was the first arrow over here? K1. down here, and it's a little k, -1. And what's this one? K2. Okay. So if I look at the Km, what is the Km telling me? What is K-1 plus K2? K-1 and K2. What are those talking about? This is telling me how much ES is breaking up, right? How about the bottom one? What's the bottom one telling me? ES formation. Have you seen this before? What have you seen this in? Affinity, right? This is affinity. Because all we're looking at is the rate that something is going away and the rate that something is forming, right? So Km is really what? KD, it's affinity, right? So this is a constant. And does that make sense that affinity is a constant? It's something that's specific for the enzyme and the substrate, right? Okay. So that's one big one. Okay. Now we're going to combine Km with our concentration of our substrate and our Vmax. What was our Vmax? Our maximum velocity of the reaction, right? You could add as much substrate as you want, doesn't matter, that's as fast as it's going to go. Okay, so if we look at this on the graph that they were talking about, right? In order to find your Michaelis-Menten constant, right? What do? You find your Vmax and you find 1/2 of your Vmax. But is Km determined in velocity? No, Km is what? It's a concentration, right? Just like normal KD, it's a concentration. So you have to find your halfway point, find your kinetics of your particular enzyme substrate reaction, drop down and find out what is that substrate concentration where the reaction is proceeding about half as fast as it possibly could. And that gives you your Km, right? So even if it is the same, as long as it's the same enzyme, the same substrate, no matter how much substrate you have, your Km is unchanged. How about Vmax? If I change my substrate concentration, does my Vmax change? Yes. What if I change my enzyme concentration? Will my Vmax change? Yes. If I change my enzyme concentration, will my Km change? No, it's a constant, right? So if you change concentrations, you do change the velocity of your reaction, but you do not change the concentration at which you have half the maximum velocity. Yeah? Okay, I think that's a really good stopping point. What you got? This one? Yeah. Okay, good. We'll stop there. Because we'll convert it to line revert next time. And then, no, I redid the calendar. I redid the calendar. So it pushed back your homework. It pushed back your exam by 1 day. Yeah.
I just want to say thanks for all those little curved arrows. I enjoyed that.
The curved arrows?
Yeah, the mechanisms.
Oh, yeah?
Yeah, I enjoyed that a lot.
That was good?
Yeah.
Yeah.
No, seriously, I'm not joking.
Oh, thank you. Don't forget exams. If you haven't checked your exam, come and see your exam. Oh, yes. Thank you. Thank you. Thank you. Oh, look, I'm going to put it in my folder so that I'm like, why is that in my folder? I'll remember. You too. Yes, you too. All good?
Wait, you said to go on Wednesday now?
Yeah, it's moved back to the Wednesday because I added that extra day and I didn't change the calendar.
Okay.
Y'all okay with that? Y'all gonna survive? So y'all got a different crew for lab today. It's a good crew. All of my crews are good crews.
Well, do you have any snacks?
Do I have,
You could go to the vending machine.
I got snacks in my office.
What kind of snacks?
I got 9-year-old snacks.
I'm okay with 9-year-old.
Come on. I gotta stop by and get my bag and stuff. So you can come check out my snack.
Forgot to put them in.
Oh, what am I doing chat? I'm trying to end.